So we have done replication where we have seen that a DNA is copied, a daughter DNA is copied, taking the template, mother strand as a template. From the replication, we moved on to transcription, where we saw that based on one strand as a DNA template, the strand which reflects the sequence of the promoter in the 5' 3' direction, that strand is taken as a template DNA strand, and on that strand an RNA is synthesized in the 5' 3' direction. And this is the primary transcript. And we also have studied that the primary transcript is then processed. And in case of eukaryotes, there are 3 or 4 post-transcriptional processing that takes place. And finally, we get a mature transcript. Now, before getting a mature transcript, we get a mature transcript of mRNA that we studied. When we are talking about central dogma, we are talking about the pathway that is from DNA, the information is being carried to RNA, and from RNA, the information is being decoded in the ribosome by the ribosome to produce a polypeptide. So when we are talking about central dogma, we are talking about the life of mRNA. So an mRNA is produced from a DNA, and the mRNA is then moving from the nucleus to the cytoplasm. And within the cytoplasm, the mRNA will be trapped, as you can see, will be trapped or targeted by the translational machinery to produce a polypeptide. So that's central dogma. So from DNA, we are getting so from the DNA, we got a desired mRNA. And from the mRNA, we are now getting a desired polypeptide. So this is the basic concept of central dogma with which we started, and as was proposed by Crick in 1956. However, the central dogma was later on modified. Apart from the mRNA that can be produced from a given segment of DNA, they have shown here, this picture shows here the varieties of RNAs that can be produced as a result from a DNA segment. They can be snRNAs, rRNA, ribosomal RNA, transfer RNA, and pre-microRNA. Since we are concentrating on the translation machinery, we will restrict ourselves to 3 categories of RNA. The first category is the mRNA, which contains the genetic codes, which will be recognized and accordingly will be weaved into a string of polypeptides. Apart from that, 2 other RNAs are also essential to facilitate the translation process. The 2 other RNAs in question are the ribosomal RNA, which is essential in the process of translation, and the second one is the tRNA, which is also essential in the process of translation. So when we talk about translation, these particular 3 RNAs are very important. mRNA is actually carrying the genetic code, it's actually carrying the information of the DNA. The tRNA and the rRNA in the process of the translation machinery will interact with the machinery and will help in production of the polypeptide. For tRNAs, to be in a very simplified manner, it has been shown here that when we talk about polypeptide, it is nothing but a string of amino acids. So you can see, so the tRNAs, what happens, what role do they play? Depending on the code present on the mRNA, accordingly, they pick up the corresponding amino acid, and you can see here, where did my cursor go? Just a second. So these are the pool of amino acids you can see here. So depending on the code present here, the tRNAs pick up the amino acids. So they are charged with amino acids, and these charged tRNAs then enter the ribosome, as you can see here. So the tRNA has a vital role here playing here. And in case of ribosomal RNA, the ribosomal RNA also plays a big role, specifically the ribosomal RNA of the smaller subunit of the ribosome, the 30S subunit, which recognizes the mRNA in a defined manner. So the ribosomal RNA plays a big role in recognizing the mRNA and thereby facilitating the sitting of the ribosome at the correct position on the mRNA. So this is in a nutshell what we are going to study. And before entering into the main topic of translation, as I said, let's talk about amino acids. Now, we all know that there are 20 amino acids, and the 20 amino acids can be divided into acidic, basic, neutral, polar, and the second category, and neutral, apolar, neutral, polar. So there in nature, we have 20 amino acids being divided into acidic, basic, neutral, non-polar, neutral, polar. The property of the amino acids depends on the R group present attached to it. So if we have an acidic R group attached to it, it's acidic amino acid. For example, aspartic acid and glutamic acid, and just see what is the abbreviation used. ASP, glue. So these are the abbreviations used and the symbol used. Specifically, this we will be studying this one later on in gene regulation. So see, the symbol used for lysine is K. Okay. When we'll be studying gene regulation, we'll come back to lysine once more. So just remember the symbol used for lysine is K. Then when we have basic charge R groups, then the amino acids we get are the basic in nature. One of the well-known basic amino acids are the lysine, arginine, and histidine. And these three amino acids do play a big role in gene silencing. Okay. Just gene silencing, gene modulation. Now we're coming to neutral non-polar, that includes tryptophan, phenylalanine, glycine, alanine, valine, isoleucine, leucine, methionine, and proline. And we have the neutral polar, which includes tyrosine, serine, threonine, aspargine, glutamine, cysteine. So these are the standard 20 amino acids found. And as I said, when I started this class, so this was the central dogma. Replication autocatalytic. From a DNA template, a daughter is synthesized. Transcription heterocatalytic. From a deoxyribonucleotide template. On a deoxyribonucleotide template, a ribonucleotide is being synthesized, hence heterocatalytic. And the process transcription. And based on the information present on a ribonucleotide, a polypeptide chain is being synthesized. So protein and RNA are also of different chemical nature. Hence, again, the process is heterocatalytic and is known as translation. This was initially proposed, but later on with the discovery of reverse transcriptase, it was seen that on an RNA template, a DNA can be synthesized, which is known as a cDNA or the complementary DNA. And so this is the modified central dogma. Now, this is something these concepts are something you know. So just a second. Now, these are what happens is that how is a polypeptide formed? So this is amino acid 1 and this is amino acid 2. We are having a carboxylic group here, you can see, and we have an amino group here. And as you can see, a peptide bond is formed between the C and the N with the liberation of water. So you can very well see that a peptide bond is being formed between C and N with the liberation of water. And so when this is basically a dipeptide, it's made up of 2 amino acids. So there is always an amino terminal or N-terminal tail of a protein, and there is always a carboxy terminal end of a C-terminal end of a protein. This is only a 2 peptide molecule, but in this 2 peptide molecule also you can see it's a dipeptide, that there is an amino terminal end and there is a carboxy terminal end. Now coming to the nature of the genetic code. What was said was, when the 4 basic bases of the DNA were known, they are nothing but A, T, G, C. Now the question is that actually the scientists faced during that time is that how does A, T, G, C, the simple codes, give rise to such complex polypeptide chains? So it was a string of experiments. We're not going to get into the details of the experiment. The basic idea was that if I take any two bases, say I'm taking any two bases. Okay, sorry. I'm taking all the four bases, and if I consider all the four bases and combination of any two bases, so they will combine, see, all the four in combination of any two, AT, TG, GC, CA, TC, so combination of any two. So all the four bases, combination of any two. So 4 into 4, that gives 16. So if any two letters specify one amino acid, then we are getting 16 amino acids. But what we just now saw is that in nature, there are 20 amino acids. So obviously, a combination of any two bases is not giving rise to a single amino acid. So that combination is not working. So the next approach was that if we change the combination number, where did my cursor go? So if we now have A, T, G, C, and these bases are now taken in combination of any three. So there are four bases and any combination of three. So 4 into 4 into 4, that gave 64 combinations. Now, if we see that in nature there are 20 amino acids and we are having 64 combinations. So these 20 amino acids can be accommodated in these 64 combinations. So it was assumed that a combination of any three bases actually codes for one amino acid. It was experimentally proved that is the relationship between the codons and the amino acids was extensively proved by Nirenberg and Hargobind Singh Khorana in 1968 using a cell-free protein synthesizing system of E. coli, where they proved that indeed any three bases in a defined combination codes for one amino acid. So this partner of these three bases is called as one codon. So one codon is a combination of any three bases that is actually coding one amino acid. Now, if we come to the concept of the genetic code, one thing should be kept in mind that even though it was the experimental evidence of Hargobind Singh Khorana and Nirenberg that provided us with the experimental documentation that yes, three bases makes one code, it was hypothesized in around 1960 by Francis Crick, Leslie Burnett, Sidney Brenner, and Watts Dobbins that the genetic code is triplet in nature. That is a set of three nucleotides, a codon in mRNA code for one amino acid in a polypeptide. But the exact relationship of these 64 codons, as I said, the combination can be 64 combinations. So the exact relationship of these 64 codons to the 20 amino acids was done by, was experimentally proved in the laboratory of Marshall, Nirenberg, and Khorana, who shared the Nobel Prize in 1968 for their work. Now, when we talk about this particular beautiful genetic code, which is unique and which is universal among all organisms, see how the code is written. Now, you should always remember that the three triplet code present in the mRNA is equivalent to one polypeptide. So keep in mind that in mRNA, instead of T, we have a U. So when the genetic code is being represented in a pictorial format, it is a representation of the codes bases present in the excuse me, mRNA. Don't confuse it with the DNA, because actually it is the information of the mRNA that has obviously been copied from the code. That is obviously being a copy of the coding strand of DNA, but it is the information of the mRNA that is being actually converted into a polypeptide. So remember that when we are seeing a genetic code, we are seeing the picture of mRNA. Imagine an mRNA in the back of your mind. So all the thymines, instead of thymine, will be getting uracil. Keep that in mind. So now, how do we see the genetic code? So you can say this is a checker box of 16 squares. So we have first row, second row. So we have a first row, second row, third row, and a fourth row, right? And we have a first column, we have a second column, we have a third column, and we have a fourth column. The first row is for uracil. The first column is also for uracil. The second row is for C. The third row is for adenine, and the third column is also for adenine. Just a second. So we have a third row of adenine and a third column of adenine, and we have a third row of guanine and a fourth row of guanine and a fourth column of guanine. So this is how the rows have been divided, and this is how the columns have been divided. Now, each box, this is one box. Each box, as you can see, is again divided into 4 rows. The first row contains U, the second row contains C, the third row contains A, and the fourth row contains G. So this is how the the boxes have been organized. So each box, again, as I said, is divided between 4 rows. So in the first row, we write uracil, second row cytosine, third row adenine, and fourth row guanine. So this is how we are going to read the genetic code. So when we start reading the first one, see how it is read. U for the row, U for the column, and U for the first row. So this is U, so we read this one. First base, second base, and now we have our third base, U. Let us take any other box. Let us go for a random order. So we choose this one. Let's choose this one. Okay. So this row is for A. So we read A, first base, A. See, this column is for C. So C is the second base. And then we have these 4 bases. We read the first row. The first row is U. So we have A, C, U. So however you read it, the first base is for the row, the second base is for the column, and the third base is for the internal rows, and accordingly which row you are reading. So this is how the genetic code is read. So once we have identified how to read the genetic code, now let us look at the property of the genetic code. The genetic code has some defined property. The first property is it is a triplet code. As I said, each triplet base codes for one polypeptide. So it is basically, and these 3 triplet bases are known as one codon. So each mRNA codon that specifies an amino acid in a polypeptide chain consists of 3 nucleotides. This is the first property of the genetic code. The second property is that it is comma-free. That means it is continuous. So if we have a sequence, let us have a sequence A, T, G, C, 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 A, T, G, C. So when we start reading a code, it is read as A, T, G, C, 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 A, T, G, C, A. We are not pausing. I mean to say the ribosome will not pause. It will read the code as a continuous stretch. It will not pause. So it is comma free. It is continuous. And the 3 bases are to be read at a time, and none of the bases are to be skipped while reading it. So as I said, again, I'm reading it ATG, CCC, ATG, CAA. So this is the property of the genetic code being comma free. Third property, it is non-overlapping. What do we mean by that? The mRNA is read in successive groups of 3 nucleotides. That means that when I start reading, I read ATG, and then I read CCC, again I read ATG, and I read CAA. I have not read it as ATG, TGC, GCC. So I have not read it in this manner. I have read the 3 nucleotides. I started with ATG, and then only I entered CCC. So this is non-overlapping. Had it been overlapping, it could have been read as ATG, then TGC, GCC. So that is overlapping. But the genetic code is non-overlapping. So you finish reading the 3 bases, and then you read the next 3 bases, and then you read the next 3 bases, and this is how it goes on. So it is non-overlapping. The fourth character is that the code is universal. Almost all organisms, this is a very, we say that we are evolutionary conserved. So all organisms that have been studied have usually the same codons that mean the same amino acid. However, there are always exceptions to a rule. The code is completely not universal. For example, mitochondria of some organisms, such as mammals, have minor changes in the code, and so does the nuclear genome of the protozoan tetrahymena. So there are certain, I mean to say that it's universal with some exceptions, but these are minor exceptions. So we have gone through the 3 properties of, it is a triplet code, it is comma free, it is almost universal, it is non-overlapping. And now coming to another property, it is degenerate. So this question more or less often comes that what do you mean by the degeneracy of the codon, or the code, not of the codon, of the code. So you can see that when we look into this picture of this genetic code, we are seeing that there are 64 combinations. But in nature, we are having only 20 amino acids. So it is obvious that some combinations are, there are more than one combinations which are coding for one amino acid. For example, let's look at this picture. You can see there are 4 combinations which are coding for leucine. There are 4 combinations which are coding for threonine. There are 4 combinations which are coding for alanine. Again, 4 combinations coding for glycine. So each amino acid at times does not have a unique codon. It can be coded by more than one codon, but there is an exception. So this is the property of the degeneracy of the code. The codons, the amino acids do not always have unique codons. They are often coded by more than one codons. There are 2 exceptions to this rule. The yellow one, as you can see, this is the methionine AUG. This is unique. There's only one combination that always and invariably codes for methionine. So wherever there is AUG, methionine will only come, and that is the only combination methionine has for it. And another one is the tryptophan. Where did tryptophan go? Just a second. There it is, tryptophan. So these are the 2 amino acids where we see that there is a uniqueness to the genetic code. So one codon is actually coding for a single amino acid. But if we look into the picture of the genetic code, we are seeing that of the 20 amino acids, 18 are being encoded by more than one codon. So this property, that of the 20 amino acids, 18 are being encoded by more than one codon, this property is known as the degeneracy or the redundancy of the code. This multiple coding phenomena is called the degeneracy or the redundancy of the code. So this is one important property. Next, as you can see, if you look into the F, the first, second, third, fourth, fifth, sixth property that the genetic code has is that the code has start and stop signals. Very, very defined. So when an mRNA comes out, a mature mRNA comes out, it has a start signal, which indicates that this is the point from where the first amino acid of a polypeptide chain is being added. And a stop signal. This is the point where the ribosome will understand that there is no corresponding amino acid to this codon, and so it opens up. So if we look into the genetic code, the picture of the genetic code, once again, we have some defined start and stop signals. So you can see the yellow one highlighted here. AUG is a start signal for protein synthesis. So in an mRNA, the first AUG that is sitting in a definite orientation to a specific sequence, there is another recognition sequence. We'll come to that later on. But the AUG is a start codon, and it signifies that this is from where the protein synthesis starts. And there are stop signals. You can see there are 3 stop signals in the genetic code: UAA, UAG, and UGA. UAA is referred to as ochre, UAG is referred to as amber, UGA is referred to as opal. So these are the stop codons. Stop signals or stop codons do not have any corresponding trna and do not code for any amino acid. You can see no amino acid is written adjacent to them. And so when the ribosome faces such a sequence on an mRNA, which it is translating, the ribosome understands that since there is no corresponding tRNA to it, the ribosome opens up at that point. So, and finally, we move to the final property of the genetic code, that is Hubbell's hypothesis. Now, if you look into this particular case of leucine, you can see that the first 2 bases is same, while the third position base has changed. But since the first 2 bases are same and only the third position base has changed, all the 4 combination codes for leucine. If we look into another case, valine, the first and the second base is the same, only the position in the third base has changed. It again, all the 4 combination codes for valine. So it is evident, we can look at the picture of alanine. The first 2 bases are same. The position in the third base is changing in all cases. But all the combinations are reading as alanine. So it is evident to some extent. We are seeing a similar picture in case of proline. We are seeing a similar, see, the first 2 bases are constant. We are seeing a similar picture in case of threonine. The first 2 bases are constant. A similar picture in cases of isoleucine. First 2 bases constant. A similar picture in case of serine. First 2 bases are constant. U and C has changed. Third base changed. Similar picture in case of arginine. First 2 bases constant. The third base has changed. So from these examples which I have now circled, it is evident that the third base in the codon is able to base pair less specifically because it is less constrained three-dimensionally. It wobbles. Third base wobbles, allowing a tRNA with base modification of its anticodon to recognize up to 3 different codons. What does that mean? Let's go by an example. So you have identified from this genetic code that where we are getting that there's a same first and second base, only a change of the third base is occurring, but all the 4 combinations are coding for the same protein. Now, so let's look into the oblink concept. Now, in the process of translation, what happens when an mRNA codon is being positioned in the ribosome in its decoding center, that codon is recognized. Check out the direction of the mRNA. It is 5' to 3'. Check out the direction of the tRNA, 5' to 3', drawn arrows. So this is 5' to 3'. This is 5' to 3' for the tRNA. The tRNA has an amino acid leucine attached to it. On the basis of what? On the basis of the fact that CUC codes for leucine. So it has a leucine attached to it, and it is now bonding with the mRNA with its anticodon. So if this is known as the codon, then this is the anticodon found in the tRNA. So it is bonding with this anticodon with the codon of the mRNA. The third base in the codon is C. And just now from the picture of genetic code, we saw that if the first 2 bases remains constant and there is a change only in the third base, for all the 4 position change, we are getting the same amino acid. Now look into this picture. The C has changed to U. The problem is that even though the codons, the third base changes, but in the anticodon, the tRNAs, if there are 20 amino acids, there are 20 tRNAs. See, we have 64 combinations of codons, but there are not 64 tRNAs in the cytoplasm. There are 20 tRNAs in the cytoplasm. So there are 20 amino acids, there are 20 tRNAs. So the tRNAs need to recognize all the 4 codons for leucine. Because leucine, as I showed you, has first and second base Cu. The third base can be UCAG. So we have only one tRNA for leucine, and that tRNA needs to identify all the 4 codons for tRNA. Now, as I said, the first 2 bases we are seeing constant. So pairing between the bases of the anticodon and the codon in the first 2 cases are strong. Here we are seeing a normal pairing. We have the anticodon of leucine read as GAG. So its normal pair is CUC. So it's a normal pairing. But we have an anticodon of GAG. Now the codon of leucine is CUU. G normally pairs with C, but G does not pair with U. There's a normal rule. But since the first 2 bases have more or less paired strongly, so this pairing, the third base codon, the third base in the codon is able to base pair less specifically. It is not, it's pairing less specifically because it's at the one end and it is not so much constrained 3-dimensionally. And that's what happens. It wobbles. The base is, the pairing is not that strong. There's a wobbling, allowing the tRNA to recognize the U, and there's a wobble pairing occurring here. And even though G pairs in a very less specifically with U, we see that leucine is incorporated into the polypeptide chain. So you can see, I have given you the wobble in the genetic code. When there is a G at the end of the anticodon, it can pair with U or C. If we have C at the 5' end, means this end. At the anticodon, it can pair with G. If we have an A at the 5' end of the anticodon, it can pair with U. Again, if we have a U at the end of an anticodon, it can pair 5' end of an anticodon, it can pair with A or G. Just remember the orientation and the direction. This is the 5' end of the anticodon, and this is the 3' end of the codon. Wobble occurs, wobble pairing occurs with the 3 end base of the codon to the 5' end base of the anticodon. So this is a less constrained base pairing occurring between these two, and this was proposed by Crick and is famously known as Wubbel's hypothesis. So this is about Wubbel's hypothesis. So summarizing about the Wubbel's hypothesis, as I said, that is the 3' end of the base codon, that and the 5' end of the anticodon. That third base pairing is bit relaxed and wobbling occurs. Now coming to the universal nature of the code. For example, I said the code is universal, but there are some exceptions to the universal nature of the genetic code. So UGA is a stop codon. It's universally stop codon, but in case of bacterial DNA mycoplasma, it codes for tryptophan. We have some other unique features in case of mitochondrial DNA. UGA is again a stop codon, but in human mitochondrial DNA, it codes for tryptophan. Then we have CGG, which codes for arginine in plants, with the universal code, and its altered code is tryptophan. So these are some variations from the universal nature of the genetic code. We have a AUA in humans. This is the codon. Universally, it codes for isoleucine, but in case of humans, it codes for methionine. When we talk about nuclear DNA in tetrahymen and paramecium, we have two stop codons. These are stop codons. They are the universal stop codons, but they code for glutamine. So this is some subtle differences between the universal codes and some uniqueness that occurs in this universal code, some exceptions. And finally, with the end of genetic code, we move on to translation of protein synthesis. We'll carry this on our next chapter. So we'll stop our class today.